Our project was in pharmaceutical engineering and the optimization of a pharmaceutical formulation. Our project mentors were Sarah Koinov and Sarang Oka. I'm Danielle Copes.
My name is Janice Gu.
I'm Elizabeth McGinley.
I'm Lindsay Schell.
And I'm Tiffany Zak. So today we're going to talk to you about how flowability of powders relates to the pharmaceutical industry. We're also going to talk to you about the V blender and the FT4 Rheometer, which are machinery that we use to make our powder blends. And finally, we're going to conclude with our research and any further steps we're going to do on this topic.
So our research focused on the property of flowability, and it's literally exactly what it sounds like. It's the ability for a powder to flow. And this is really important in the pharmaceutical industry because if a powder doesn't flow well and it sticks in the machinery, that can be really harmful to the powder overall because it might no longer be homogeneous. And like homogeneous, just equal mixture throughout, and that's really important because when you make tablets, if they're not homogeneous, they could have different dosage sizes, and that's really dangerous to the human body if you have like an inappropriate amount of a certain ingredient. So the ingredients that make up drug products are the active pharmaceutical ingredient and the excipient. So the active pharmaceutical ingredient, or the API, is what provides the therapeutic effect for whatever ailment that you're experiencing. And they're generally the smaller particles that stick together more. So the smaller, like when those particles are smaller, they have more surface area, so they don't flow as well. And the API that we were using was confectioner's sugar, which models acetaminophen, which is commonly found in Tylenol. Previous research has shown that acetaminophen and confectioner's sugar have very similar flow properties because of their similar particle sizes. So it's okay that we use the substitute of confectioner's sugar in this experiment. For our excipient, we use microcrystalline cellulose pH 102, which has a larger particle size, therefore allowing it to flow better. And excipients have various purposes, such as superficial things like color and taste of the drug, but they also do more important things like improving the flowability, the way MCC does.
So the machinery that we use to make our blends was called the V blender, and it's very commonly used in the pharmaceutical industry. And basically, its shape leads to its functionality. You load your powders into one of the legs, as seen here on the picture, and as the powder all falls down to the bottom of the drum. And then the V blender rotates, and as it rotates, the powder becomes divided at the apex of the V, and this process is rotated over and over again, and it usually results in a homogeneous blend. However, since this is only a uniaxial rotation, it only rotates this way, your blend may not be as homogeneous as you want it to, depending on your material and your loading procedures.
The machinery that we use to actually test the flowability of the powders is the FT4 Rheometer. And we'll talk more about the actual procedures we use for testing later on. Right now, this is just the equipment itself. There's 3 different stages, and they each use a different blade for testing and preparing the powders for testing. The first stage is the mixing blade, which is the top picture. That removes clumps of powder in the powder. And the second blade is the compaction piston, which is the middle picture there, and that compresses the powder and removes air bubbles. And the third and final blade is the one on the bottom, and that is the Shear cell module, which is the blade that actually tests the flowability of the powder.
So when we're testing for the flowability on the FT4 Rheometer, we look for pre-shear and shear. And pre-shear is what happens to prepare to find the failure point, which is what actually leads to us finding the flowability. So the failure point, pre-shear goes through and finds a pre-shear point, which is where when the shear cell module goes into the powder and turns slowly, as more and more shear stress is applied, when a constant volume is found, that is what the shear pre-shear point is. And then from there it can move into shear, and we can find the failure point, which is where when the blade goes into the powder again and rotates slowly, when the powder breaks and like rolls over each other, that's called the failure point, and multiple failure points can give us the yield locus.
So the yield locus basically shows the points at which a powder fails. So you take the failure points from what Danielle just described, and you graph it onto a shear stress versus normal stress graph. And then you take the linear line of best fit through the shear points, and that gives you your yield locus, which is also known as the yield limit. This point here is called the pre-shear point, and that's the point of steady state flow. And that point is generally located beneath the yield locus line.
From the yield locus line, which is the blue line up there, we can determine the unconfined yield stress and the major consolidation stress, also known as the major principal stress. and this can help us find the flow function, which helps us find the flowability. So if you see the yield locus there, you can take the points tangent to it. The first one goes through the origin, and that gives you the unconfined yield stress. And the second one goes through the pre-shear point, and again is tangent to the yield locus, and that gives you the major consolidation stress.
So this is the flow function. The flow function is basically a measure of a powder's flowability. So if you have a high flow function, it means that the powder flows very well. And if you have a low flow function, it means that the powder does not flow very well. So on this graph, which is the unconfined yield stress versus the major consolidation stress, the flow function is defined as the reciprocal of this slope. So in other words, it's the major consolidation stress divided by the unconfined yield stress.
So before we started our experiment, there was a little bit of research we had to do. In order to make our mixtures, we had to figure out exactly how we need to make our mixtures. So we had to do some research into previous experiments involving MCC and acetaminophen to determine how we would make our mixtures using convection sugar as a substitute again. So we found that a rotations per minute of about 20, a fill level of 50% in the V blender, and 10 minutes of rotation would generally create a homogeneous mixture so we could have consistent testing throughout our experiment.
So in this experiment, we focused on one variable, which was the percentage of API. The other three factors that Liz mentioned, the RPM, the fill, and the blending time, were kept constant after our preliminary research. However, this could lead to a confounding of factors. A confounding factor is a third variable which can influence the relationship being studied of two other variables. So in our case, that would be the percentage of API and the flow function. So ideally, we would have individually tested the RPM, the fill, and the blending time for which combination produced the most homogeneous blend in the end. But because of the time restraints of this project, it was not feasible for us to do so.
As I mentioned before, we used the V blender to make our blends. So basically, we used 1 kilogram of powder per blend, and we basically load our excipient and our API and powder form through one leg, and this is simply to provide a more homogeneous mixture. We also did increments of 10%, increasing in 10% of API. So we go from 0% API to 100% API, and after that we would test the flowability. The amount of powder of 1 kilogram also perfectly fit approximately the 50% fill level of the V blender. And we chose 10% increments because we felt that this data set would allow us to create the best flowability graph that we had.
So once we actually have the powder mixed, we can test the flowability in the FT4 Rheometer. And as Lindsay mentioned earlier, there are 3 different steps. The first one is where the mixing blade goes down, and it just makes sure that there aren't any air bubbles or clumps of powder stuck together, making sure that the powder is consistent all the way through. The second one is the compaction piston, which presses down into the powder to a desired pressure level, which our group used 3 kilopascals, but the pressure isn't much of a factor in what's actually going on in the experiment. It's more to ensure consistency between experiments. And the final one is the shear cell module, which goes into the powder and twists very slowly. But before it goes in, we open the container and split it in half, as you can see on the bottom picture down here. And this is just to make sure, again, a constant volume, so that way every test is similar to the last one and most consistent as possible. And again, pre-shear and shear test the flowability going through, seeing what we're going to find out for flowability levels.
In our experiment, we conducted 3 trials for each different blend mixture. This was to ensure consistency within our graph. However, some data points had outlying data, so we retested them with additional data to ensure a more reliable graph.
So these are the results that we actually got from testing the experiment. We plotted the active pharmaceutical ingredient by mass percentage against the flow function, and as you can see, we got an exponential function here. And the line of the best fit was 10.168 times 10 to the negative 0.021 to the x. And the R squared value was 0.9225, which shows that there was a very strong correlation there. We had very typical results of what we expected based on prior research. The excipient had larger particles, so when there was less API, we had better flowability, which makes sense. Although on the left side of the graph up here, we had a lot of outlying data points, which was unexpected for us.
There's a couple of possibilities for these outlying data. One explanation is the environmental factors, such as temperature, humidity, transportation time, and or transportation experience and storage time. For example, some of our powders were used immediately after being produced, while others sat overnight, so this could lead to changes. Another possibility is the particle size and the gaps between the particles. The MCC has larger particle size, so there's more space. So this could lead to like different, okay. So then the third reason, which is most likely, is that the FT4 Rheometer has trouble quantifying well-flowing patterns, which, as you can see, our outline data is mostly at the points where the pattern flows best, so this makes no sense.
So just to reiterate, our goal was to determine whether or not there was a correlation between the percentage of API of a powder by mass and its flow function. And through this experiment, we determined that there was a correlation between the two. We found an inverse exponential relationship. Again, like Danielle said, the equation is y equals 10.168 times 10 to the negative 0.021x, where x is the API percentage and y is the flow function. And our high R-squared value just goes to show that we had a strong fit between our trend line and our data points.
There's a variety of applications for our research in the pharmaceutical industry. You can both design drugs with appropriate flowabilities for specific machines, as well as design machines for drugs with specific flowabilities. And this can help optimize the manufacturing process of drug products. Also, we can save costs by using a substitute instead of an API, like we did with confectioner sugar in place of acetaminophen, because a lot of API can be very expensive in large quantities. And saving the costs in the research and development stage can overall lead to increased productivity and diminish the costs, which makes the drug more affordable and accessible for consumers.
So there's definitely a lot of room to improve our research. As we mentioned before, there's factors in the blending process, such as the time of blending, the RPMs, and the fill levels of the feed blender that could have affected our flowability graphs. So we can test those factors and have graphs that consider those factors. We also did increments of 10%. So further research, we could do increments of 5% or even 1% API, and that would just make our graph more detailed.
So in addition to bettering this experiment, we can also repeat this experiment using different combinations of APIs and excipients. It's important to remember that the conclusions we drew from this experiment can only be applied to a mixture of MCC and acetaminophen or confectioner sugar, because all powders flow differently. They don't all have the same flowability properties. So if we repeat this experiment using these different mixtures, we can create different graphs for all these different powders. And if we create a large enough database with all these different mixtures, then we can perhaps find a general flowability rule that powders seem to follow, and that can be useful in any industry that utilizes powders. So as we conclude, we would just like to thank our mentors, Sarah Koinov and Sarang Oka, for helping us, teaching us all how to use the equipment, and just being really supportive.
We'd also like to thank our residential teaching associate, England Collins, for advising us about the project and basically becoming a great friend of ours.
We would like to thank the director of the New Jersey Governor School of Engineering and Technology, Eileen Rosen,
as well as the assistant director, Jean-Patrick Antoine, for coordinating and running the program.
And finally, we would like to thank our sponsors for letting us have this wonderful opportunity.
Any questions?
You had mentioned that your machine that turns only had the ability to turn one direction. I would imagine in commercial applications they have much more powerful machinery and would mix.
Yeah. There are things like the, what was it called? It's like a biaxle. It rotates like both this way and this way, so it's really good.
Much more powerful than
Yeah.
if you had to work with them. That's great. Thank you.
Really nice work. Congratulations. I have a minor question in your graph. I'm wondering, do you have any thoughts on why most of the outliers tends to cluster at the left side?
I had explained that with some of my reasons for the outline data. Like I said, environmental factors, as well as the equipment itself. It just has a hard time with the patterns that tend to be more well-flowing, like those patterns were.
Thanks.
So how much do you know about the size of the microcrystalline cellulose of the particles
think
and the
it's
shape?
150 micrometers.
Okay.
The
And
pH 102 is what indicates its particle size.
Okay. And is that uniform? I mean, how broad a dispersion? Does it
The MCC, yeah, it's
Spec-
generally
uniform?
Yeah.
Okay. And anything about the shape? I mean, are those plate-like crystals or
it's
rounded?
Do like makers of Tylenol, do they use MCC or do they use a different substance?
Well, in general, Tylenol, I'm not.
They use a lot because they have different factors, whereas we were only testing flowability. Whereas they're looking for other things. They also look for, like Liz mentioned, superficial things like color and taste as well.
There's other excipients like that have purpose, like lubricants, to help with the machinery also getting it so it doesn't get stuck. So in Tylenol, if you look at the list of ingredients, there'll be like magnesium sulfate or that kind of stuff. So that they'll have different purposes. They're all excipients though.
There's no other questions. One more round of applause.